Hello, everybody. In this lecture, we're going to talk about the basics of genetics, and then we're going to move on to some slightly more in-depth coverage of genetics in the next couple of lectures. So with that, let's get right into it. And so by the end of this module, you should be able to define genotype and phenotype and discuss their relationship. Compare and contrast the ways eukaryotic and prokaryotic cells' genetic material is organized in their genomes. And to get at that, we're going to start out with some basic terminology. So genetics is the study of genes, their functions, and how variations arise in their genomes. Which brings us to genomes, which is the collection of genetic material in a cell or virus. Genomics is the study of an organism's genome, and metagenomes is the study, or metagenomics is the study of all the genomes in a bulk sample, while metagenomes are all of the genomes in a bulk sample. We would be remiss if we didn't mention Gregor Mendel. He's the father of modern genetics. He was an Austrian monk. He worked with pea plants, and what he was doing was tracking whether or not traits are heritable and can be passed from one generation to the next. We also have Wilhelm Johansen, who came up with the term gene. It was used to describe heritable units of genetic material that determine a particular trait. And he came up with these ideas of genotype and phenotype. So genotype is your genetic makeup. So that's all the gene, that's the gene that you have for something. And that influences your physiological trait. And so that would be your phenotype. And so this idea that your genotype can determine your phenotype is the prevailing theory of genetics that we use today. So you can think of genomes as being instruction manuals. These are going to have all the possible features of an individual cell or a virus. And they're going to be made up, or they're going to be determined by your genome. And so all cells have a DNA genome, while viruses can either have DNA or RNA genomes. Now, as a general rule of thumb, more complex organisms have larger numbers of genes. And so an example of this would be E. coli K12, which is just a normal lab strain of E. coli. It has about 4,400 genes, while pathogenic E. coli 0157H7 has about 5,500 genes. Now, if we move to a eukaryotic yeast cell, they have about 6,000 genes, while humans have about 24,000 genes. Now, this isn't a perfect one-for-one. Also, don't think of humans as being the most complex type of organisms, right? There are some organisms that have much larger genomes than humans, while they may not be as socially complex as we are. They do have complex lifestyles overall. So the chromosome is what your genome is organized into. When we have chromosomes in our different organisms, what this is going to be the DNA packaged tightly, usually associated with some sort of protein like a histone or histone-like proteins that allow them to be condensed really tightly. Now, of particular note is that the number of chromosomes does not influence an organism's sophistication. So a very complex organism can have a small number of chromosomes total, as opposed to a very simple organism could have more chromosomes. It's not a direct relationship there. Eukaryotic chromosomes are linear, and they are housed in the nucleus. In this image, you can see the histones, which allow the DNA to get wrapped around them and helps prevent them from being tangled up. Prokaryotic chromosomes are going to be the circular chromosomes. They're usually only going to have one, while some can have two or three chromosomes. And they have these histone-like proteins, which allow the DNA to coil around them in the nucleoid region of the cell, but they are not membrane bound. Bacteria can also have extra chromosomal DNA called plasmids. And so what we have over here is a transmission electron micrograph of this small circular plasmid. These are not part of the primary chromosome. They usually carry beneficial traits to that organism, but they are not required for life. Plasmids are going to be smaller than the genome. The genome is where the majority of the genetic material is kept. These plasmids are extra chromosomal, which means that they're outside of the chromosome. And like I said a moment ago, they tend to have extra genes associated with them. So these could be things like antibiotic resistance or metal resistance or metabolism of a special carbon compound or some sort of virulence factor. Here is a table kind of comparing prokaryotic and eukaryotic genomes. It's useful to be familiar with these differences between these two types of organisms. It's all the stuff that we just talked about, but it's worth reviewing this as well. And with that, you should now be able to define genotype and phenotype, as well as to compare and contrast prokaryotic and eukaryotic genetic material and how it's stored. With that, I hope you enjoyed, and I'll see you soon.